Perfect.
Great. Thank you. Yeah, Daniel, very good job, and good job with the Zoom bombing as well. So taking care of that. So we're going to talk about some things. So another theme of this week is development, and so we'll be talking about development mostly in embryos instead of in larval stage animals. But we still have this. Get that out of the way. Okay. Yeah, so this is some of the work. It's a collection of work that we've done over the last 5 or 6 years in our group here. This is the DevoWorm group, and this is our website at the top there and the hyperlink. And we're affiliated with the OpenWorm Foundation. We do a lot of work with data that's secondary data or tertiary data. So it's like data generated by other people. And we analyze those data. We come up with, do different analyses of the data. We also build models. We do a lot of modeling and a lot of machine learning. So this is the an example of our group here. What we have in terms of data, we have, we have mostly microscopy data at this point, but we have some molecular data, and as you will see, some reproductive data as well. But this is all made public, and we believe in open data, so we are very attuned to that. But we also do a lot of analysis on our own of different imaging data and other things. So we have developed some segmentation and processing pipelines. We do some modeling and simulation as well. We do a lot of quantitation and developing quantitative models. So we have two different sort of sub-interest areas. One is the DevoZoo, which is a collection of open data. And another one is the DevoWorm AI/ML. So AI/ML stands for artificial intelligence and machine learning. So we have a strong interest in that as well. And I won't show any of that work today, but keep in mind that that's something we're interested in. So one of the things I'd like to point out before we start is we talk about models. And in this talk, we'll be introduced to some models that seem pretty abstract. But this is one thing we have to remember when we talk about models in general, and that is that we want to avoid spherical cows. And what that means, it's one of my collaborators in the group. He talks about spherical cows as oversimplifying a biological system. So development actually is interesting because it's actually a sphere, and it's actually a spherical cow at some point. Embryos are spherical. But over time, they become these asymmetric organisms. And so the question is, in embryology specifically, you want to go from that spherical organism to a highly asymmetric organism. And so some of our work has been surrounding this process of developing asymmetry. How do things become more asymmetrical over developmental time? Associated with this is this idea from John von Neumann, where he criticized a lot of modeling. This was 50 or 60 years ago when he made this observation. But he made this observation. The quote is, "With 4 parameters, I can fit an elephant, and with 5, I can make him wiggle his trunk." And so you can see over here, here's some elephants, here's some equations, and you can build these models in graphical form. And the lesson there is that just because you can model something that looks like it's realistic doesn't mean that it is. You can build a lot of spurious models with data. But that's something I wanted to point out, just to be skeptical of models in general, but also something we kind of take seriously in our group. And so I think what we're trying to get at is we're trying to build a simulation of an embryo, or we're trying to build something that is generalizable and maybe is synthetic in some way. So there's been some work on synthetic embryology, and it's been a little bit, here and there. So one example is groups that have developed what they call artificial human embryos. And so these are cell cultures where they combine progenitor cells and organizer cells. And they grow these things in culture, and they assay them for gene expression and other molecular markers. And so you can see on the right, this is an image of what they've produced. They're able to look at higher order patterns in these embryos. So in this case, they're looking at Wnt and nodal signaling, the combination of them and how they affect the embryo, how they sort of explain what's going on in the embryo as it's developing. But that's sort of the one type of approach, synthetic embryology approach. Another is a new paper that's out from a group where they've taken the ascidian, which is a sea squirt. It's a sea animal, and they have looked at the early embryo. And the early embryo in sea squirts is much like C. elegans, and I'll talk about that in a bit. But they've been able to take each cell and map molecular data to each cell and build a virtual model. So those are the types of, that's the state of the art and what we're trying to do. So you can see it's sort of scattershot, and some of our attempts are maybe a little bit less integrative than we'd like, but that's basically where we're going. But we also have this problem of representation, and I think we think of like biology as being as descriptive as possible, but in our case, we have to think about this at different levels of representation. So each thing, from behavior to genetics, has its own like sort of level of explanation, level of representation. And our modeling mainly focuses on two things: behavior and phenotype. It's not that we don't care about the molecular data, it's that we've excluded it from our models, and we may add them later. But to think about this representation problem modularly like this, I think is quite effective. What we've done now in our group is we've come up with this 5-dimensional data structure. So in this case, we have this, this on the left is a point cloud of like you take a cell, you segment, and you find centroids in a 3-dimensional space. So this is the product of cell tracking. And so we rely on this database out of Riken, which is a systems biology oriented database where they have a lot of shared data sets, and they've processed them and put them in standardized formats. And so we've been able to work with these data a lot of the time. So we've been able to build these, we smooth these point clouds, and we can identify individual cells and build models from that. So we have a 5-dimensional data structure. 3 of these parameters are from this spatial model, and 2 of them are from the lineage tree. So we look at things over time as cells divide and differentiate throughout the lineage tree. And we have this I parameter, which is, it's spatially independent. It could be based on the ordering of the lineage tree from anterior to posterior. It could also be some contextual information, but that's the way we approach this. And so again, this is a 2-dimensional array of cartoon cells. This shows you kind of the position, and then there's some time and context that we can add to as well. So given that, we've done a number of pieces of, we published a number of papers using this type of approach. One is to compare C. elegans with this sea squirt, this Caenorhabditis intestinalis, which is a single species within this order. And the thing about Caenorhabditis is that they have, like C. elegans, this deterministic type of lineage tree. And so you can map the cells over divisions. I mean, originally the embryo looks like this, but you end up with, this is the larval form, and then the adult form is much different than this even. But at this stage in the embryo, you have this, you can track each cell as you go along. And so it's ripe for a comparison with C. elegans. And so what I'm talking about here is actually a larger theoretical issue having to do with the mosaicism of development. So in development, in embryonic development, that is, you have 2 sort of, most organisms, most animals exist between 2 different poles, either the mosaic form or the regulative form of development. So mosaic development is what we have in the sea squirts, and this is the adult, or the C. elegans. And the developmental cells have a predetermined fate. So you can trace them through a lineage tree pretty well. You know every cell is going to be in roughly the same position. Every cell has a similar identity coming from the mother cell, and so we can, that's a pretty reliable signal in terms of reconstructing the tree. On the other hand, you have regulative organisms, which like fishes or mice, and they have a highly variable lineage tree. So cells are regulated by chemical signals, by morphogens and things like that. And they take on fates depending on the embryo. So every embryo you look at is slightly different in terms of its distribution of cells and what they become. People have tried to reconstruct lineage trees in this way using DNA barcoding and things like that, but that's not something I'm going to get into today. Just suffice it to say that most organisms exist along this um, continuum, and that mosaic embryos can lead to regulative adults. So in C. elegans, you actually see that a bit with the post-embryonic nervous system that generates cells in the post-embryonic uh, phenotype, and then you see plasticity, which is regulative. And so we're comparing the two organisms here. We're comparing C. elegans and Ciona intestinalis. And there's some interesting things you can do. For one, you can look at the relationship between cell volume and division time. So this is from our data. We have, these are different from the 5-dimensional paradigm I was talking about before, but this is using additional data. This is looking at the trends between cell volume and division time. And so you can see that we can characterize this, and it's characterized differently in each species. We can also look at new relationships, though. So we can actually confirm old relationships and characterize new relationships. And so in this case, we have a case where we look at cell volume versus the lifetime of the cell. So it's a slightly different relationship. But we can see that we can pull out cells. We have one sort of group of cells that follow this trend line, and then another group of cells that follow a different trend line that are sort of act as outliers along the cell volume. So they're a little bit bigger in terms of cell volume, and they live, a variety of lifetimes here. This is the founder cell for the D lineage. This is P3. So you can identify individual cells in the embryo that deviate from the mean of the embryo. So we can do that. And we can do that again in Ciona intestinalis. So in C. elegans, we can do this, and we know kind of what the cells are. We also know this in Ciona intestinalis, although the cells aren't labeled here. But the point of this is that we can identify trends in the embryo. We can identify like emerging structure, things that maybe are, different about different parts of the embryo. So for example, in C. elegans, you have certain sublineages that go on to form like germ cells and other types of specialized cells. And so we can maybe identify the precursors of those differences here at this stage. But we're also looking at building a meta-embryo. And so the meta-embryo, we can again take these point cloud data, and we not only have this for C. elegans, but we have this for Drosophila, and we have this for zebrafish. And so we can look at all these embryos at different stages with the same type of tracking data, smoothing it out, and putting it into this framework. And we can do a lot of things with that. We can look at the distribution of major events. So another thing we've done is sort of map out major events. We mapped out sort of the emergence of cells in the connectome, but very early, not even like before hatch, which is before L1. This is when the cells are first emerging. And then comparing those cells with the adult. So we have a publication on this where we've identified when these cells first come up in development, and they come up between 265 and 400 minutes. This is when the neurons first start to differentiate. And so we also look at things like, different other events in this as well, not just the connectome. And so we have these things that we can characterize. So we can characterize cells in the embryo as they're differentiating and dividing. We can identify very early stages of the connectome. And so we have all of this information, and we're able to characterize, and we've characterized it in a number of publications. So we can look at these distributions and major events. These are different cell families that are differentiating in different stages of development. So we have this by minutes. So this is the first from 200 to 300 minutes of embryogenesis, when some of these cell families come online. Like these are hypodermal cells, and so they come up fairly early, but there are other cells that come about later. And we can look at these distributions and compare them between cell types. So between interneurons and neurons, interneurons and hypodermal cells. So we can look at the distributions of those in development. And then not only can we say things about that, but we can work towards simulating these in sort of a generic embryo. And so this is some work I've been doing with someone where we've been talking about building this sort of synthetic or meta embryo that is sort of independent of any organism, where we can take all the insights we learned from, like, say, C. elegans or Ciona intestinalis and apply them to this model. And so we can do this by, looking at sort of like how we might build a lineage tree from what we've learned, but also from like certain mathematical sort of assumptions. So in this case, we're looking at generating a lineage tree from a Poisson process, and seeing if that looks something like what we see in nature. We also have new methods that we've developed. So this is something called information isometry. And so for this group, it's going to be pretty abstract, but I'm going to walk through it a little bit. So say we have two lineage trees, and we base one on, like, say, on the actual lineage tree that you might observe sort of from microscopy, the standard lineage tree, the cells dividing and their location in the embryo. And we have another lineage tree that's maybe based on molecular information. And so if we have those two lineage trees, they're not going to be the same in terms of their ordering along, say, the bottom of the tree, the tips of the tree, or even the internal nodes. And so how do you compare? We may be able to compare, say, like something built from a molecular assay versus something built from like a anatomical assay, and see what if there's any information in that. And so developmental cell lineages contain multiple sources of information. We can use those multiple sources and compare them. And so what we do is we use what they call a Hamming distance, which is a type of information that looks at, like, we basically turn these nodes into binary strings, and we calculate the difference between the strings. So in this case, we're looking at the difference between two trees, and we're looking at the binary strings, and then we're calculating some numeric difference between them. So in this case, if we compare two trees, and I just generated these sort of not at random, but I just picked an example, we have, in this case, this node here in one tree is the node over here in another tree. So it's a distance of 4. In this case, this node in one tree is over here in another tree. So it's a distance of 1. And so you can look at these, you get these numbers, and it's a Hamming distance between the two trees. And you can actually plot them in a bivariate space. So in this case, we've plotted it out so that you can look at the sort of the evolution of these distances. So for example, you start off at a distance of 0, you go to a distance of 1, and then 3, and then 4. And you can maybe see how different these lineage trees are over the course of cell division and differentiation. And so these are what they look like as visualization tools. And you can see that they look like pearls on a string of different colors, but these different colors represent different subtrees. So if you think about like the comparison processes swapping subtrees, that's exactly what you see here. You have different subtrees that are swapped, and you see the distance here. And so the idea is to visualize those differences, but also we can make other calculations on them as well. And so in this case, just to make sure that we're not looking at random noise, we actually generated a bunch of random trees and compared C. elegans to this over the course of development. This is based on lineage tree levels. So this is the very early embryo, and we looked at the average Hamming distance, and you can see that there is a real biological signal here, at least in terms of C. elegans versus some randomly generated tree. And so this is a comparison of subtrees. And then eventually, we want to apply it to other organisms. But the problem with, say, C. intestinalis is that C. intestinalis exhibits fourfold symmetry. So how do you deal with that type of complexity? It's not what we see in C. elegans at all. And so a potential solution might be to use these more sophisticated geometric shapes to characterize these changes. Finally, I'm going to talk about some empirical science that we've done with collaboration with other labs. So this is Nathan Schroeder at UIUC and his group that we worked with on this. This is changes in fecundity due to larval starvation. So in this set of experiments, we worked on capturing data on L1 starvation. And so people who aren't familiar with that, if you take a bunch of eggs and you treat them with SDS and you hatch them, they're synchronized and they hatch, and you can get them to L1. You can put them in 15 degrees in a buffer, and they can survive there for up to 2 weeks. But the thing is that they starve as they do this, so they're putting a lot of energy into surviving. And so when you take them out and you put them on a plate, and at L1 stage after they've hatched, they start to grow, but they start to grow at a different rate. And a lot of things happen that they've had to put a lot of their energy into sort of surviving. A lot of things happen in their lifetime that are far different from, say, a wild type or one that hasn't been treated. So in this case, we're using a wild type N2 worm, and this is the case where the worm has not been starved. So this is the wild type where it's not been starved, and you can see it forms a peak at the second day of adulthood. So this graph is how much reproduction is it doing in adulthood. So this is like 1 day after we plated it. So this is early adulthood, and this goes out to 5 days. You get up to you have a peak at 2 days, and then it declines. But in the example that's starved, you have this delay in terms of when it starts reproducing, and it doesn't reproduce as much. So you can see there's this case where the L1 is just allowed to develop normally, and then the L1 is starved and then allowed to develop. And so you can see this difference. And these geometric shapes are actually taking like the number of offspring per day and then building these shapes. And one reason we want to do this is to make calculations on the area under the curve. So the difference between these two curves, we want to characterize them in this way. And so we can characterize this area under the curve and say things about the difference between starvation and non-starvation. This one obviously is reproducing much more, but then this changes later in lifetime. So they're producing more offspring later. And so you get actually 3 different components to this. You get skew, which is where the shift in peak reproduction shifts forwards or backwards in time. You get delay, so you get this a little bit of reproduction here at 2 days, and then it goes up. So you could say that reproduction was delayed, although that's a little trickier to characterize. And then this kurtosis, which I'm referring to as kurtosis, it's really just this change in peak population size. So the population peak, when it's producing most of its offspring, decreases. And so we did this in wild type, but we also did this for a number of mutants. And so we did this in AAK1 and AAK2 mutants. And AAK1 and AAK2 are well known for their, if you put them in any sort of stress, they have different effects on the longevity of the worm and their physiological function. So the AAK1 and AAK2 show deficits in their and their metabolism. And we didn't know, there's been some work in the literature, but it wasn't very well quantitatively characterized. So that's why we picked these mutants. And you can see for AAK1, there's a decrease in the kurtosis, and there's no real skew in terms of the reproductive peak. In AAK2, however, there's a huge difference in that the AAK2 worms that were starved, or the AAK2 mutants that were starved, have this like very late in life reproductive boom. I don't know what else to call it, but there's some sort of protective effect maybe later in adult life that happens. I don't really know how to characterize it other than that, but suffice it to say it's very different from the form that wasn't starved. And then for the double mutant, which we created for some of these experiments, there's a shift in the reproductive peak. It's slightly accelerated here. It's earlier in terms of the number of days, and it's a little bit, the kurtosis is a little bit lower, but it's pretty similar to the unstarved form. And so those are the types of things you can do with mutants and with starvation, and we've done this in a preprint here, and we've tried to quantitatively characterize them. So we can apply this to a number of different types of mutants. So this has been a whirlwind of a tour, but we would like to invite you to visit our group and perhaps join if you're interested. So we have a lot of contributors. The way our group works is we have contributors that come and go, and they can contribute as much or as little as they want. We have different sort of badges that people get as they participate. and that's just on our website by the names, but it gives people a sense that, it just shows the diversity of contributions we get. We have people affiliated with Google Summer of Code, which is a nice internship program. We have the SSBD database. We collaborate with them. We, of course, work with OpenWorm and with INCF, which is a organization for neuroinformatics. And so here are our contributors. There are more contributors at the website. And finally, we have our weekly meetings on YouTube, so you're welcome to join. Consult the website for more information or email me. So I will stop there.
All right, thank you very much. It was very informative.
Thank you.
If there is any question, please write them on the chat. So I have a question. Like when you were for like a part of your talk, you were mentioning that you were using microscopy data and molecular data. Is molecular data like information like transcriptomes, RNA-seq data? Because this is getting more and more as like everyone is sequencing, right?
Right. So we have worked a little bit with molecular data. We've not done a lot. I mean, there's a lot out there, but the problem with open data, and this is a more general point that we deal with in our group, is that it's people provide data, but it's not really in a form we can use all the time. So that's why we've invested in sort of segmentation and processing data. We have that that sort of investment because we need this to be able to like put the data sets together to use them in different ways. We're not focusing a lot on molecular data, but we're interested in moving in that direction. It's just a matter of finding the right opportunity.
All right. While we're waiting for question, I have another one.
Okay.
So when you're doing your tree comparisons, so you like, do you highlight new biological information with that, like new lineage of, or like link with molecular data and cell lineage?
Well, what you have in the comparisons is you can take like the standard lineage tree, and what you get is you get the cells that divide, and they have this deterministic sort of tree organization, topology, whatever you want to call it. With molecular data, or maybe sometimes you would compare like size data or something, you'd reorganize the tree, but it wouldn't be like a wholesale reorganization. In C. elegans, it's going to be like, you're ordering it from, say, left to right or right to left. You're switching the nodes from the mother cells. And so that means that you're switching the daughter cells. So sometimes you're swapping subtrees. And so that's based on a different form of information than what we've traditionally built our lineage trees on. And so it can get kind of confusing. And of course, in the regulative embryos, you get even a worse problem with that. But this is an example, very simple example, where we can say, well, there's a reordering of the tree at each level. We can characterize that and see what, maybe there's some information in there. Maybe the cells divide and we can track those cells, but there's molecular information so that there's stuff being expressed in one cell sublineage versus another one. And it doesn't really match what we think of when we think of the lineage tree, but there might be information in there that's important.
All right, thank you very much. Like I don't see any question yet, but I think I have a last one. When you compare the volume, like your volume plot of the cell, like with the early one, where you have like you compare the one between the C. elegans and the Drosophila, which were like roughly the same shape. But was I correct to see that for the zebrafish, it seems that like there is a plateau on the upper part of the bubble?
Yeah, the zebrafish embryo is a little bit different in terms of its shape. So what we're capturing with those point clouds is largely the shape. So it's I think it's at the top. It's there's a little bit of the cell tracking doesn't really get at that top part very well, but it's just a thing that happens in zebrafish. It's a little bit unique. So like every embryo is a little bit unique. It's not just this massive cells, although it looks like that. It's it's actually a lot different in reality.
Okay, thank you. Well, I'm guess we went a little bit out of our time window, and there is no question yet. But if anyone has question, please feel free to ask them on our Slack channel. For the people which are still around, my apologies for the Zoom bombing. We're going to try to fix that for next week. And Bradley, thank you again very much, you and Daniel, for 2 very good talks this week. And we will see everyone next week for some microbiome stuff. Thank you.
Thanks.
Have a great day.
You too. Bye.